Good day, Homo sapiens. Good day. Welcome back to studying Prescott's Microbiology. And this is Ray Microbiology. So, it's been some time, quite some time since we last went live, right? So, the last time we studied, by the way, how are you guys doing? I'm doing perfectly fine. I was just busy. So, by the way, the last time we studied the secretion pathways, or in general, you can say in the last streams in the chapter 3, we studied the internal structures of prokaryotic cells, right, and their functions along with that, and their mechanisms if included, right. It wasn't that complex, but it was still, the BS level and related to microbiology, right. So today's topic is the components external to the cell wall. Components external to the cell wall. Wait, let me just move them to the side, and let me just check if my live is working, because just few minutes ago when I went live, it wasn't working because the volume was cut off, right? Like my mic was not connected, so I don't want to repeat that mistake. Okay, it's working. So let's just go here. So we are studying components external to the cell wall. So today we will be covering two main things, right? That is the outer covering, outer to the cell wall, even in gram-positive and gram-negative cells we have peptidoglycan layer, or you can say the lipopeptidoglycan type layer, right? In gram-negative, in archaeal cell walls, of course, they have their cell walls, right? We already discussed that. But there is another outer layer that is prior to the cell membrane, outer to the cell membrane in even the cell wall, and that are the capsules, slime layers, or S layer, or collectively you can say these two are collectively the glycocalyx, and this one is just a unique piece, a different type of information you might get. Now another thing that we are going to discuss today is the pili and fimbriae. Guys, this is very too common knowledge, right? Like we should know that, because it is present in almost not all of them, but like it is present abundantly. So we will see what the pili is, what the fimbriae is, and what the capsule, slime layer, and S layer. Then there are other things that still comes outwards of the external, and that is the flagella. You will have heard, but I am skipping flagella today because flagella is a detailed topic, its mechanism, its structure, and specifically, it is very common, and it has a lot of functions. So as you can see, this is extremely detailed topic. Alright, so I am skipping that. And then we have chemotaxis, etc. Like that is, just leave it. It's up for after things, but it is not the external structure. So we are just studying the external structures to the cell wall. So guys, don't worry, it is very simple. I'm going to cover it in a few, you can say. Wait, is this girl moving or not? Yeah. So first of all, what is capsule? Guys, we eat capsules, right? Like it has a covering that saves our important stuff. The same case with the bacteria. There comes bacteria that have an extremely solid outer layer, right, prior to even the cell wall. It means that even the cell wall is not sufficient for them. They are greedy. They want to be even more protected and even more resistant. So they develop an other mechanism, and other structure that further strengthens their boundaries and mechanisms, and that are capsules, slime layers, or S layer. To be honest, their first function is to protect them, right? And by protecting means, of course, it protects them from osmosis, right? Because even though their cell walls and cell membrane has permeability, specifically, specific levels of permeability, capsules just locks that and just won't let them be destroyed by the osmosis or by that kind of things, right? Similar will be the case with the slime layers and S layer. They are all layers. Some are made from proteins, some are made from peptidoglycans. Now, the other thing that the capsule, slime layers, and S layers do in mechanism wise, not structural wise, but mechanism wise, function wise, that is evading the immune system of the host cells. we have attack mechanism as well. Like if the bacteria enters our body, we have attack mechanism, antibodies, and different kinds of proteins that we attack them with and destroys them. So bacteria with complex cell walls, or things like that, they evade that. Like for example, gram negative are more problematic than gram positive because they are very resistant to dying. They are difficult to die. So when capsule comes, so our immune system now can't recognize them because they mostly covers the whole identity of the bacteria. If our body can't even recognize what the bacterial cell is, how is it gonna attack? Like if you don't know you are being attacked by a bird or by a plant or by bomb, how are you going to deal with that? Like you don't even know. The same case with capsule. It hides their identity and evades the phagocytosis process, right? The next is the slime layer. Same slime layer is the same as the, what do you call it, the capsule. Like the function wise it's different. Now the S layer is also the same. Now the third thing that the capsule does is, wait, what was it again? Oh gosh, it skipped my mind. What do I do? What skipped my mind? There was something. Okay, whatever it was, let's move further and we will know. I am going to read all of this below. So as you can see there, if you want visualization, do you see this outer layer, the pink color? This is being stained by the dye, counter dye or whatever. You can see it in a compound microscope as well. Like you don't need electron microscope for this because the capsules are very thick things. So you can see it visibly. It's actually thicker than the cell walls most of the times because it is a capsule. So this is how you see it generally. This is how you see it. Okay, so this is of pneumonia and this is bacteroids. I think this one is stained differently, right? I think it's stained differently. That one is differently. Oh, so that's what do you call it? Dark field microscopy. This is light field microscopy. You might have it. And this is fluorescent dye. Means it is emitting light, as you can see. Now, if you want to see, this is intestine. This diagram, it is intestine, right? And these are bacteria. Even though I don't know what they are arrowing towards the glycocalyx, I'm sure they mean this, these kind of, these lines coming out of it, like this is the whole capsule of it. I'm sure you guys cannot see, but the zigzag or the distortion around it. This is actually capsule, but it is not that much clearly visible. I mean, if you focus and these lines, it's not visible. Let's forget it. Who am I kidding? So these are intestinal tissues. These are bacteria, and there is a glycocalyx, means the outer layer around it. Here you can see it clearly. Now, the pili and fimbriae, right? So both are usually used interchangeably. I think in our country, pili is used very commonly. And maybe in some countries fimbriae is used very commonly. Like it's people usually just use a general term for those two, but these two are different. Maybe not drastically in structures, but their functions are different. So first difference is, first let's know what the pili is. These are the filaments like structures, filaments, right? And they surround the cell. Wait, let me show you. Is it here? Here, these thin like structures or whatever. Let's look at this one. Like is these kind of things. I know this is structure of different things, but here it's like these kind of filaments like things. And fimbriae are actually this much in amount, like it's extremely high in amount in numbers. But pili, these are just from 9 to 10, 5 to 10 maybe. Like it's not high in number. And the other thing that differentiate between these two, other than the number that they are present in, is their function. Fimbriae are usually used for attachment and maybe motility, right? Location changing, movement. pili are usually not for that. Like, don't mix it with the flagella, right? Flagella is also for motility, and flagella are actually more centered towards motility than attachment. Fimbriae actually have role in attachment. Now, if we have flagella for motility, we have fimbriae for attachment, then we have pili for transfer of DNA. It's actually more used as a tube that will transfer DNA from one bacterial cell to another. Let me draw it for you. Wait, come on, yeah. For example, this is your cell. There are, oh my god, my drawing. Just think there are a lot of fimbriae, okay? A lot of fimbriae, oh my god. Let's rewind it. Specifically bacteria, what kind of bacteria is cocci don't have usually these. So this is bacteria. Now if we have a pili on that pili, just consider it as by the way, it is more thicker in diameter than the pili. If fimbriae are thin filaments, then the pili is thicker filament, and it is longer and thicker in diameter than the fimbriae. Now pili, just consider the cell has pili, the other cell will also have pili or won't have it. This pili will connect with this bacteria, and there is a specific type of DNA that we call plasmids, unicellular DNA. And of course it has its own DNA as well, like RNA or whatever. It has its own genetic material, but it has an extra genetic material that we call plasmid. And pili are used, it's a tube like, pili makes a tube like structure and connects another bacteria with itself, and then it transfers this genetic material, it duplicates this and transfer it through this tube to another bacteria. And now this bacteria will also have plasmid, so it's kind of used for transfer of DNA mostly. So that's why it is called as sex pili. Like in our generation, in our mitosis in humans, we also transfer genetic material, right? And that's that's why we have sexual reproduction. That's what we call that. But in bacteria that don't occur, and so this is the process that I just show you. This is known as conjugation, and this is one type of sexual reproduction in bacteria. As you can see, it's transferring genetic material, even though it's not by meiosis like us, but it still occurs, and it is with the help of these pili. But remember, pili are not for just transferring the genetic material. They can act as motile or attachment purpose, like they can do that as well, but it's rare, as compared to the conjugation. But fimbriae are high in number, and I mean it's like their second job, or not second job, like it's like breathing to them, attachment and movement. Now wait, does this one have diagrams? Okay, so this is shown as S layer. Let's forget it. These are fimbriae. Oh, my crappy drawings, they actually had a good pictures. Why did I tortured you guys with my drawing. Here you can see these small, I'm sure you guys cannot see it because it's so mixed with the backgrounds. So you guys can see where does it go? So these small structures around it, these are fimbriae. Now it's just the reading time, so let's just read, okay? First, let's get ready. Wait, which one was it? Yeah, let's get ready first. Are we ready for the reading? Okay, we are ready. Come on, can we move down? Reading will take only 5 to 10 minutes. Let's see if I can do it in 5 minutes. Don't worry, I'm not going to skip anything. Let's move my other windows to the sides so I can read. Okay. Now, prokaryotes have a variety of structures outside the cell wall that can function in protection, attachment to the objects, and cell movement. Several of these are discussed. Now, the first one is capsules. Some prokaryotes have a layer of material lying outside the cell wall. This layer has different names depending on its characteristics. Okay, so they are saying prokaryotes have a layer outside of the cell wall. Some prokaryotes, okay, not all of them. Now, it is named after these depending on its characteristic. When a layer is well organized and not easily washed off, it is called as capsule. Means the most resistant one and the most organized one is known as capsule. Now, what is slime layer? It is called a slime layer when it is zone of diffuse. Means when it doesn't have clear boundaries and it doesn't have organized material and that is removed easily. Means if it is diffusible and if it is unorganized, it will be easy to remove because it is not structured well, it is not made well. When the layer consists of a network of polysaccharides extending from the surface of the cell, it is referred to as the glycocalyx. So anything that is made up of polysaccharide and that is extending out of the cell wall, that is known as glycocalyx. So in this case, the capsule and the slime layer comes under the category of glycocalyx. A term that can encompasses, what's wrong with my cursor? A term that can encompasses both capsule and slime layer because they usually are composed of polysaccharides. However, some slime layers and capsules are constructed of other materials, for example, bacillus synthesis. So now, if you exclude the glycocalyx, they are saying that there are other layers that are made up of different things. Unlike, for example, what name did they use? Bacillus synthesis, right? Like in bacillus synthesis, the outer layer is made up of protein. See, the proteinaceous capsule composed of poly-D-glutamic acid. We know what glutamic acid is, right? It's an amino acid. Amino acid, whatever you call it. Capsules are clearly visible in the light microscope when the negative stains or special capsule strains are employed. Okay, capsules can be invisible if you don't stain them because it's jelly-like structure. Have you ever seen starch when it is not cooked or when it is cooked? It becomes a transparent-like thing. So polysaccharides or you can say proteinous structure can also become that. Glycocalyx or whatever these are. So capsules are clearly visible in light microscope, guys. Even in light microscope, just think they are so larger and thicker. But it needs stain. Although capsules are not required for the growth and reproduction in the laboratory cultures, like we don't need the capsule. Like even if you remove capsule from the bacterial cell, it won't change their activity much. It's like armor. Like if we wear armor for our protection, and if you remove them, it doesn't change anything for us. Just it reduces a little bit of protection, but our functions don't matter because it doesn't work in our functions. So the same case is with them. So they do confer several advantages when prokaryotes grow in their normal habitats, and that is they make them resistant, they make them strong, and that's kind of advantage they give them. They help pathogenic bacteria resist phagocytosis by the host phagocytes. Like if it is a pathogenic bacteria and its environment is living in the host, so how is it going to survive in the host mechanism? Because host is going to attack them because it's not going to recognize these pathogenic bacteria and they will be considered as enemies. So capsules actually let them resist the phagocytosis. Phagocytosis is the eating of the foreign materials by the cells, immune cells. By the way, solid material, okay? If you eat solid material, if the cells eat solid material, that's phagocytosis. If it eats, or you can say drink liquid material, it is pinocytosis. Now, Streptococcus pneumoniae provides a dramatic example. Now, this is the example of a pathogenic bacteria that avoids the phagocytosis, or you can say avoids the immune system. Now, how when it lacks a capsule, it is destroyed easily and does not cause disease. Like if this species doesn't have capsule, or if you remove the capsule, it won't cause disease because it will be killed easily. But whereas capsulated variant quickly kills the mice, but when it is capsulated, it is not easily destroyed. That's why it can cause disease. Because it's surviving and it is pathogenic, of course it's gonna cause disease until you kill them. Capsules contain a great deal of water and can protect against desiccation. Exactly, it helps in the maintenance of osmosis. It can retain water because It won't let them leave. They exclude viruses, most hydrophobic toxic materials such as detergents. Okay, and not just for phagocytosis, not just the osmosis, but they also make the bacteria safe from the viruses attack and the toxic materials, because they won't be able to go into the bacterial cells because of the double triple security. The glycocalyx also adds the attachment to the solid surface, including tissue surfaces in plants and animal hosts. Okay, so they have adhesion properties as well. Gliding bacteria often produce slime, which in some cases has been shown to facilitate motility. Motility means movement. So if you are not sticky, bacterial cells usually have receptors, so it's kind of reacting to everything. But when you have capsule, you have fewer receptors, so it is easier for you to slide down, right? Like in case if the layer is slime, so that's why it's called a slime layer. Many prokaryotes have regularly structured layer called an S layer on their surface. In bacteria, the S layer is external to the cell wall. Of course it is, because we are studying the outer layer things. The S layer may be the only wall structure outside the plasma membrane. Oh, okay, it's not outside the cell wall. It's outside the cell membrane. Come on, these ads don't even let me study. Where were we? Okay. See? Oh. In bacteria, S layer is external to the cell wall. Excuse me, then why they are... Oh, in archaea. Sorry. In bacteria, S layer is outer to the cell wall. In archaea, the S layer is outer to the plasma membrane. Okay? Because some archaea... lacks cell wall. The S layer has a pattern, something like floor tiles, because it is highly organized, so it makes a structure, and it is composed. Wait, guys, we have seen the diagram, right? Look, this is like the floor structure. Look how it's like the beehive or something. Like it is. They are not floor tiles, but if you see, it has a structure and it is arranged in a pattern, and then it forms a network like structure, and it is so strong. It is I'm sure it is more stronger than the capsule and the slime layer. In gram-negative bacteria, the cell layer adheres directly to the outer membrane. It is associated with the peptidoglycan surface in gram-positive bacteria. Because the S layer is outer to the cell wall in both gram-positive and gram-negative bacteria. So in gram-positive, since the outer layer we have the peptidoglycan, and S layer directly attaches to that. But because the gram-negative one don't have peptidoglycan as an outer layer, it's lipids, right? It's lipopolysaccharide. The outer layer is that. So that's why it doesn't adhere directly into within the gram-negative, but it adheres directly within the gram-positive ones. It may protect the cell against iron and pH fluctuations, osmotic stress, enzymes, and other predaceous bacterium. Vibrio. Again, it's an outer layer, so it's further secure and it's further permeability, so that's why it can protect cells against iron and pH fluctuation, osmotic stress, enzymes, and other bacteriums, etc. Just think of it as a protector. The S layer also helps maintain the shape and envelope rigidity of some cells. Of course, it's gonna help in rigidity. It can promote cell adhesion to surface. Again, the same thing with the capsule and the slime layer. The S layer seems to protect some bacterial pathogens against the host defenses, contributing to their virus. S layer isn't as evasive of the immune system as capsules, but it still might, help the bacterial strains fight against host immune system. Now, pili and fimbriae. Okay, I thought I was going to end it in 5 minutes. What the heck? Many prokaryotes Many prokaryotes have short, fine hair-like appendages that are thinner than flagella. Of course, flagella are actually high in diameter, large in diameter, and long in length. These are usually called as fimbriae. Fimbria is a singular. Although many people use the terms fimbria and pili interchangeably, we shall distinguish between the fimbria and sex pili. Okay, so they directly named the pili as sex pili. It's not like that, but mostly pili usually act as sex pili, so that's why they are differentiating between these. Now, a cell may be covered with up to 1,000 fimbria, but they are only visible in electron microscope because of their extremely small size. Now, the first difference was fimbria are high in number and they are extremely small in size. They are slender tube composed of helically arranged protein subunits and are about 3 to 10 nanometers in diameter and up to several micrometers long. So the diameter is 3 to 10 nanometer and the arrangement is not linear, it's helical like DNA, of proteins instead of nitrogenous bases. At least some types of fimbria attaches bacteria to the solid surfaces such as rocks and in streams and host tissues. Fimbria act as attachment material, some type of, not every type of. Some types act as motility factors, some types act as attachment factors. And maybe some types act as evasive factors. Fimbria are responsible for more than attachment. Type 4 fimbria are present at one or both poles of bacteria. Guys, I'm not sure what these type 4 or type 3 fimbria are. We are gonna Google it at the end of the stream. For now, let's just get along with it. Like there is a type 4 fimbria and it is present on both poles. So this is a type of fimbria. Just as we said that oh, F4 S M here, why? I mean, welcome. You are here because you want to study, of course. So they can add an attachment to the objects and also are required for a twitching motility that occurs in some bacteria such as Pyrogenosa, Neisseria gonorrhea, and some strains of E. coli. Okay, so they are saying there is a type 4 fimbria since we said that there are different types of fimbria. And they are saying type 4 act as attachment ones and twitching motility. Means they act both as attachment fimbria and the motility fimbria. And they are saying these are present in strains like Pyrogenosa, Neisseria gonorrhea, and E. coli. Movement is by short, intermittent, jerky motions of up to several micrometers in the, it's like up and down, up and down, up and down. This is jerky motion of these fimbria. There is evidence that fimbria actively retract to move these bacteria. Yeah, that means they are saying that there is evidence that we have seen these fimbria actually act these ways to move these bacteria. I mean, we have seen them act as a motility factor. We have seen already the diagram of fimbria. Type 4 embryo are also involved in gliding motility by myxobacteria. So this is another example of gliding motility. These bacteria are also of interest because they have complex life cycles that include the formation of fruiting bodies. They are talking about the myxobacteria, but we are talking about fimbria, so let's skip that. Many bacteria have about 1 to 10 sex pili. Okay, now they are moving towards the sex pili from fimbria. Are you AI? No, I'm not AI. Does AI talk like that? Gosh, people these days. These are hair-like structures that differ from fimbriae in the following ways. Pili often are larger than the fimbriae. We know that. They are just two differences from the pili and the fimbriae. That is, fimbriae are smaller, thinner in size, and higher in number. Pili are, did I say fimbriae? Those were the characteristics of fimbriae. And pili are smaller in number, but larger in size. Around 9 to 10 nanometers in diameter, they are genetically determined by conjugative. We already have seen, right? And studied what the conjugation means. Plasmid and are required for conjugation. Some bacterial viruses attach specifically to their receptors on the sex pili at the start of their reproductive cycles. Now these are viruses, okay? So not does the pili can act as a receptor as well, right? Okay. So pili can act as receptors for targeting the bacteria. So this is enough for today. I can't believe I take this long just to read few topics or whatever. So today we have finished the capsule slime layers, and S layers means we have finished two factors, the outer layer and the pili and fimbriae. The next topic will be the flagella and motility. I'm not sure after how much time I will be alive, but still we are done. So how do we end this? What kind of drink what? Water? Okay, do you have any questions? I can talk for 2 minutes or 3 minutes. Or after that I will be ending the stream. Wait, let me enlarge that. Drink some water. I didn't see the water. Where is the water? By the way, what is this emoji with okay? Fish? Fish? Your fish might need water. Wait, how do I enlarge that? Okay, so I will be ending stream in 3 minutes. It's 16 for me now, 17, 18, 19, I guess. By the way, thank you for offering water. So where was it? Okay, let's enlarge you. And by the way, did you study or are you just interested because it's avatar? Okay, what? I thought you were AI. What the? Why is the AI dominating everyone these days? Not everything is AI. By the way, I think this is a compliment. You know, humans are made up of errors. AI don't make mistakes that much. Should I take it as a compliment? If my video popped on in your feed, that means you study a lot. Because my whole YouTube channel is mostly on study videos, you know. Do you study microbiology or some other subject? I mean, it's related to biology also. Definitely not. Then how did it pop up? That means YouTube algorithm is not working fine if it is showing my videos on on people feed that don't study maybe. Oh, you're a BSc student? Nah. Oh, that's a good thing. Are you at the start or at the end? It's a 4 year. Believe me, I just came out of the BS. This is ridiculous. You know, so much ridiculous. It tires you out. That's why I wanted to start the channel because I mean there is nothing interesting of studying. Are you S? What is S? I don't know. Is it a shortcut, a new shortcut these days? Of course, I'm a microbiology student. I've done my BS. Or near to completion, you can say. Anyway, I think the time's up. Oh, okay. So I think the time is up and I should end because the video is getting long. But still, whatever. Let's just talk. Do you have any concerns about your studies? You can ask me. I might be of help since I have been through this. It's not dancing. I do have an emo and what do you call it? A button for dancing, but these were not dancing. I was running. And I don't know what is this one. Mocking someone? Okay, this one is I think. Wait, let me show you. Oh my gosh, dancing on a study video. Wow, this one is I think. Tina. Wait, why did I stop? Anyway, I think I should end the stream. Goodbye, good luck. Study well if you have to study, play well, and thank you for commenting, thank you for talking. And I hope you even heard a word I have said about studies. Of course, I don't usually talk in the middle of the streams, so it's a good thing that you came at the end. So yeah, good luck, goodbye with your studies and everything, and I'm out.